This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode 64 for January 1st, 2010. Happy New Year, everyone. I'm Vincent Racaniello, and joining me today on New Year's Day, 2010, is Alan Dove.
Yes, indeed.
From Western Mass, I guess.
That's right. That's where I am. That's where I guess I am, too.
Welcome. And also Skyping in from Oregon is Rich Condit.
Hello. Great to be here again.
Thanks for joining us as well. For both of you on a New Year's Day, I'm sure you have better things to do.
What could be better than this?
Actually, you're right. What could be better? What about you, Alan? What could be better?
I don't think I have anything better to do.
Would you be working otherwise?
Probably not. Nobody's in the office. It's not like I could interview anybody. So
it's a good day to do a TWiV.
Yeah.
So we have to start getting used to saying 2010.
I'm going to say 2010.
2010. That's a good one.
Eventually, we've got to switch over to 20 whatever. So this would be the year to do it.
That's right. We used to say 2009, right?
Right.
Not 29.
Right.
Interesting. 2010. Cool. Well, I'm sure I'll write 2009 on the checks for a while.
Oh, sure.
Although I don't write too many of those anymore. Today we're going to review the top stories or 10 virology stories of 2009 because we like them for various reasons. I don't know if they're the top stories, but they're things we've talked about in the past year. And we'll go over them and add something and tell you why we think they're neat. And as usual, if you have questions, send them in.
Yep.
And of course, what else would be the first story except swine origin H1n1 influenza?
Oh, has that come up?
I think so. We did talk about it a few times.
Yeah, it sounds kind of familiar.
Way back in March and April, we started talking about it. I pulled up an old blog post I wrote just to show everyone that I can be as wrong as anyone else. This was a post I made on April 24th at virology.ws called New Swine Influenza Viruses in Humans. And I started out by saying, a new strain of swine influenza virus has recently been isolated from 7 persons in the U.S. Is it time to break out the swine flu vaccine of 1976? So I went through a long post explaining why I didn't think it was of any consequence. And then in the end, I said, swine influenza viruses probably routinely pass among humans and swine. In this case, they were detected as a consequence of heightened surveillance. Gerald Ford won't be rolling over in his grave over this incident.
Oh!
Yeah, well...
I listened to this listening to back issues of TWiV.
How does it sound when you listen to it that way?
It's terrific. It's like some sort of, movie unfolding. Okay, because it took you guys by surprise.
It did,
and you can see the thinking evolve and the interest evolve. It was very interesting. It's fun to listen to. It makes a good series.
Now maybe we should package them all together and sell a CD for $1.
Right.
There you go.
Yeah, well, this one caught me by surprise because I just thought it was another swine virus. And in retrospect, they are isolated now and then from people, but this was the real deal. This was a reassortant that had everything it needed to transmit among people. Got genes from a couple of different pig viruses, human influenza viruses, bird influenza viruses, and boom, it transmits, and most people in the world were not immune. So it became the first pandemic strain since 1968, which was before I started doing virology, and you guys as well.
That was the year I was born.
You were born in 68?
Yeah.
Cool.
It was actually before my time, I must say. Well, now, wait a minute. Is this true?
68?
68. I just started working in a lab in 68. I was in college, I was just starting to work in a lab.
Yeah, 68, I was in high school. I hadn't gone to college yet. So it was before, but I do remember the pandemic, and we've been waiting a long time. And when I went to get my PhD in an influenza lab, Peter Palesi's lab, we were all talking about when the next pandemic would emerge. And it's been a long time. How many years is that? 70, 39, 41 years.
Yeah.
That's higher math.
Yeah. New Year's Day math.
Yeah, it's exactly.
Right.
It's the
It's a long
champagne
time.
is still kicking in there.
Because the previous one was 57, 68. That wasn't very long.
9 years.
I don't remember the
11 years,
57. The 57 one I don't remember. I was too young. And anyway, so this virus has spread very quickly starting in April. It went around the world very quickly following the air routes. It's a pandemic. It's infected millions and millions of people. It caused a big peak of disease throughout the northern hemisphere in the summer, ending about August. And then down south in the southern hemisphere in their winter, they had a peak of disease. And then up here, and starting in September, it made another peak. And interestingly, now it's kind of leveled off.
Now, what I think will be interesting to see is if we get a regular seasonal flu wave coming through now.
Yeah, now typically it should be starting this month.
Yeah, because the
January.
H1n1 wave came through early for a flu season.
Right.
So we could still get a regular flu season.
So are you thinking of this virus doing it or the old seasonal strains?
Hard to say at this point because this virus, so many people have either been exposed or vaccinated. I mean, the vaccine's now finally rolling out the door at a pretty good clip.
Right.
So it seems like this virus would be at a distinct disadvantage, whereas the seasonal people may have gotten vaccinated or they may not, but there's a lot of people who aren't paying attention to that. So there's probably a large susceptible population.
Yeah, I mean, it could be.
So certainly the data so far says that this virus has effectively displaced the seasonal flu. Is that a fair statement?
Yeah. I'm looking at CDC numbers for week 51 in the U.S. 3,900 specimens tested. Only 154 were positive for flu, and 78 of those were novel H1n1. 76 were not subtyped, but they're probably all the same. And then there's nothing of H3 or seasonal H1, 0.
Now, why would this one be able to displace viruses that don't share the same immunity?
That is the question. Whereas Dick would say that's the 64-cent question. Oh, no.
I don't understand it. It's showing up at the timeline is different, right? I mean, this is, as you pointed out, not flu season.
Right.
It's typical for pandemics because they behave differently. And, the regular seasonal flu is controlled by lots of factors, immunity, temperature, humidity.
See, that's why I think we could still get a seasonal flu wave.
Yeah, but we haven't seen any of those viruses, so I have a feeling they're gone. So if we do have a wave, I think we should have a wave now in January.
Right.
But I think it's going to be the 2009 H1n1 virus. Why the others have gone away? One of the ideas, of course, is that old name of original antigenic sin, where a response to the current virus actually gives you a memory response to some previous strains that you've been infected with. And
right.
Hence, preferentially, in fact, and hence those are rid of. But it's not clear that that's the cause. I mean, that we talked about that on a recent TWiV, actually.
Yeah.
I mean, it's a reality. It's a real phenomenon, but how it plays into what happens in circulating viruses, nobody knows. Interesting question.
I'm looking at CDC numbers here on deaths and cases. This is from April to November 14th, 2009. 47 million cases, obviously the U.S., 9,820 deaths, and that works out to be like 0.02% mortality.
Right.
Now, that's certainly no higher and perhaps even lower than the seasonal flu. Is that correct?
That's right. It is. I mean, it happens to be lethal in a different kind of population than seasonal influenza targets. This is mostly younger people, whereas typically older people over 65 are the ones susceptible. But here we think that that age group has some immunity because of previous exposure to related viruses in the 50s or earlier.
Right.
At least that's one of the ideas.
And the other thing I don't have a sense for at this point, maybe you do, but is whether there's going to be any real excess mortality as a result of this infection. I don't know if it's appropriate to talk about this year or next year during the flu season, but ordinarily associated with a pandemic
Yeah.
is some spike, some excess mortality over and above the normal
Sure. Well, even with seasonal.
mortality with influenza. And it's interesting. We've been so on top of this infection from the very beginning. It's interesting to see whether that perhaps has an effect ultimately on keeping down the excess mortality.
Yeah, and if this virus has successfully displaced the seasonal flu, and we don't get a second wave of the seasonal flu, then I would think we would end up with a season with decreased mortality, which would be ironic in a pandemic year.
Right. Well, I mean, it could be a combination of the extensive spread and the immunization,
right,
which is limited. I would be very surprised if there were not a second round of flu this winter.
Do you have any idea how what the vaccine coverage has been like?
I don't know. I don't have the numbers right with me here. No.
That's a still breaking story because for their supply problem was so acute for so long. That people simply, even people who could get it. I think just a few weeks ago there was a statistic that a third of the people who wanted it couldn't get it. So then now it's all rolling out the door, and the vaccine is becoming widely available. So no telling.
I've read in some articles in Canada they feel that they've now immunized enough of the population to stop epidemic spread. I don't know if that's true. That's from an epidemiology viewpoint. I'm looking at pneumonia and influenza mortality, excess mortality, as you said, Rich, in the U.S. ending 12/12, and there's been a spike of excess mortality since about week 42. Not as big as the excess mortality, for example, in 2008, which was a seasonal year. And but the difference here is if the next graph I'm looking at number of flu-associated pediatric deaths. Now, pediatric deaths by flu are well documented. Anything under 18 actually has to be very well documented if it's caused by flu. And there's a lot more deaths in that age group this year than there were before. So that's starting at week 21, which is back in the spring. So that's one difference, and it's, again, probably because of the low immunity in that population. But this has been a very interesting development. We've watched it in real time, and we've had all kinds of data on the virus, on the sequence. We've watched the vaccines being developed. We've had all kinds of controversies about their safety, their potency, adjuvants, and the public has really been involved as a result of this medium that we are using, the internet.
Right.
And it's good and it's bad, as we've said before. Anyway, this is a developing story still. We'll see. We'll be covering it on an ongoing basis for sure in the next year. And it's pretty much dominated TWiV for 2009, I would say.
Yeah.
Right? We talked about it on just about every show. Most of the questions are about pandemic influenza. And I can't imagine any other virus infection that we know of at the moment doing that. Can you?
Well, first thing that comes to mind is SARS. I wonder what TWiV would have been like when SARS broke. I mean, that was over pretty quickly, wasn't it?
But
still, that was
but
in some ways
the interest in it was intense. Yeah,
it was.
Right,
yeah,
because we knew less about it.
On the other hand, podcasting and internet use were a lot less widespread at that point.
Right.
Yeah. So this is the first real widespread infection that has jumped on us, and we have all this internet accessibility. But you're right, SARS would have been a good one. Well, something else could come up. We never know. And we'll see. But I have a feeling it's going to be flu this year and then all the other viruses. And the others are interesting to us. We love them, but the public doesn't seem to get as engaged as they do. Maybe because flu covers everything. A lot of infections, some deaths, vaccines, whether you should be vaccinated or not. And that's probably why everyone gets engaged.
So H1n1, the H1n1 story was really the first killer app for the internet that didn't involve porn.
That's right. I'll tell you, at the peak of the season, well, I shouldn't say the season, there were some months where my blog was getting 10,000 hits a day.
Wow.
But you can tell what people are looking for, and it all had to do with flu.
Right.
And H1n1, and the search terms, they were all influenza or adjuvant driven. Very interesting. I think and the same with the podcast. I think we've had a lot of interest driven by flu, and I hope that a lot of those people remain.
Sure.
Because if you can learn about all viruses, which is what we try and do here, you'll be better ready for anything that comes along, including flu. That's my view, anyway. So that's it. That's one story number one.
We won't spend that much time discussing all 10 of these stories. But
Well, I don't know how much time we got. We got 6-7 hours.
Yes, probably you and I could stay for hours, Rich.
Yeah.
Right.
But that's fine. Yeah, that was a big one. Next one is Xmrv and chronic fatigue syndrome.
yes, indeed. This is a cool story. The whole Xmrv thing is, I think, a really cool story of 2009. See, there
Good job.
trying to use it in retrospect. So Xmrv is this virus that a couple of years ago nobody had heard of, and now it's starting to be connected to all sorts of chronic conditions. And we had someone on the show who works on it, didn't we?
Yeah, Jason Rodriguez. Yep. He has a paper out this month, by the way, in the Journal of Virology on Xmrv. Yep.
And in this case, so Xmrv, Jason's work centered on its involvement its correlation with, I should say, prostate cancer, right?
That's right. Yeah. In fact, he was on the show, talked about prostate cancer.
Yeah, and then next thing coming up was this study that found again a correlation between Xmrv infection and chronic fatigue syndrome. Chronic fatigue is a somewhat controversial diagnosis. It's a diagnosis of exclusion, where essentially if you don't have anything else but you have this set of symptoms, then it's thrown into the chronic fatigue basket. But the interesting thing was that they found this virus associated at a statistically significant rate with people who had chronic fatigue. It occurs at a much higher rate than among people who don't. So then, of course, the immediate conclusion that a bunch of people leapt to, including, I think, one chronic fatigue clinic that was involved in this work, was, this is the cause of chronic fatigue syndrome.
Yeah.
Which was something we discussed and dissed on the show, as I recall. That just because the virus correlates does not mean that it causes. And in fact, I think the fact that it's now associated with two completely different conditions, chronic fatigue and prostate cancer, to me suggests that it's not causal. It suggests that it's a bystander, that it's something that's showing up in patients who have immune systems compromised by other factors, whether that's chronic fatigue or prostate cancer, or I would expect you would find it in people who are clinically depressed. And I would expect you would find it in people who are immunosuppressed for other reasons, like transplant patients. So those would be my predictions, and I hope somebody's looking at those and seeing if that's in fact the case.
The association with prostate cancer has not been consistent, right? The chronic fatigue syndrome study did not come up with the same association with prostate cancer that the original prostate cancer study did. So that's a little tenuous.
Yeah. And in fact, another prostate cancer study that was done, I think, in Germany found no connection between Xmrv and prostate cancer.
Right. Yeah, I think for both syndromes, for both diseases, we need to still look some more. We're not done. Xmrv, I mean, if you look at the discussion on my blog and also on TWiV, whenever we talk about it, many people with chronic fatigue post comments, and, they're very frustrated because for many years, no one has understood the disease, and now they're grabbing Xmrv, and many of them are already going out and getting antiretrovirals. And we should say that some of the antiretrovirals that work against AIDS also inhibit Xmrv replication.
But those are mean drugs.
AZT.
Yeah.
It is.
Right.
Big side effects. And in fact, a paper recently has been published showing that AZT inhibits Xmrv replication. But I just don't know if that's going to be the answer. I have a suspicion there's, as you said, there's something else here.
Yeah.
And people are looking. I know others are looking, and the thing to do is to really take a big sample of CFS serum and do microarrays and look for all viral sequences and see what's there. Because here, this was a directed search. The person who discovered this, Judy Mikovits, in CFS patients, had an idea that this virus would be involved because there was a linkage between the prostate cancers and CFS patients with this RNA cell mutation. So some of the chronic fatigue people had a mutation in RNA cells. So she said, let's look for this virus. So
right.
She found it, but that doesn't mean there aren't others, right?
Yep, you tend to find what you're looking for.
I'm surprised that the microarray experiment hasn't been done. It seems so obvious.
I know. Yeah. I know. I agree with that. It should have been done. I'm pretty sure it's ongoing.
Right.
And this is going to be a big story in 2010, I think.
Yeah. And as I think I mentioned before, one thing that keeps coming up with chronic fatigue is people say, well, they're is it a real physical illness or is it a psychosomatic illness? And I think what a lot of people simply don't realize is that a psychosomatic illness is a real physical illness.
Right.
This is not a distinction that we really need to be drawing.
No, it's clearly an illness. An interesting question. Many chronic fatigue patients develop cancer. And a paper just came out showing that this retrovirus Xmrv doesn't directly induce transformation. So it doesn't have an oncogene in it. But it can induce oncogenic transformation in cultured cells. So it does have some ability to transform cells. So again, we don't know how that's related, but it'll be interesting to hear that out.
And you have the same issue with causality. I mean, do you know if these people are developing cancer because their immune systems are suppressed, or if they're which way the causal train runs?
Right.
So this is related to a murine virus.
Yes.
Yes.
I don't understand how it's acquired. Is it actually a zoonosis, or is this adapted to humans and humans are passing it around?
Well, I think it was originally a zoonosis, and then now it's passed from human to human.
Right.
Okay. My understanding is that most of the samples are very similar in sequence, most of the human viral samples, so suggesting they have a unique origin or a few origins. And there's not repeated infection from mice to people.
And is there any evidence from the phylogeny as to when this jump happened?
Not that I know of. It's very close to the mouse virus, incredibly close, so there's not a lot of variation going on. So I think we need more samples and more comparisons with mouse viruses to be able to do that.
Yeah.
I also wonder whether there could be vertical transmission. I mean, theoretically.
Yeah. It could be.
Sure, it could. I was watching a program the other day. I think it was a discovery program on Darwin, and they're talking to a guy who studies mice. He goes out in the wild and he traps them and he sequences them. And I was just thinking, he has a trap. He pulls the mouse out with his bare hands and he's rubbing the fur and explaining, fur patterns and evolution. And I'm thinking, you ought to be wearing gloves.
Yeah.
Those guys have Xmrv or the mouse equivalent.
Or hantavirus.
Yeah.
You name it. They got lots of stuff.
Stuff we don't even know about. So.
Right.
Xmrv. Yeah, that's a good one. Number 3 is the Thailand HIV vaccine success, in quotes.
Yes, success has to be in quotes anytime we're talking about HIV vaccines, unfortunately. So this was the result. Came out, when was this? This was just in September.
Yeah.
So this was a trial that was 16,000 volunteers in Thailand, and they used a combination of 2 vaccines. And this was a really interesting trial because, in fact, a lot of people, and I will say I was one of them, I didn't publicly state anything about this, but I was very skeptical of this approach when I saw the structure of it. They're taking 2 vaccines, neither of which had worked. In fact, I think one of them, the trial had been stopped because they found the people in the vaccine arm were getting infected at a higher rate than people in the control arm. So they took 2 failed vaccines and gave them simultaneously. And what they found was that this combination worked, which I certainly wasn't expecting, and I think a lot of other people in the virology community were not expecting. Yeah. But it's a very, very interesting result. So it cut infections by about 30% in the people who got the vaccine compared to those who got a placebo. And this is, in the AIDS world, that's a phenomenal success. Now, if we're talking about something like a polio vaccine or a measles vaccine, you'd say, well, 30% it didn't work. But this is a virus that nothing else has worked before, and now finally there was a success story, and that was really jaw-dropping.
Yeah, that we talked about back in September on TWiV 51, and Rich came on to tell us about the vector, because this is a canary pox vector.
Yeah, right.
It was used to vector one of the antigens here. Yeah, the numbers were very close, and in fact, that in September was a preliminary release of the data, and then subsequently it was discussed at an AIDS meeting in France, I believe, and the numbers got even closer than they were in this study. Still statistically supporting protection, but very small out of 16,000, 20 or 30, a difference of 20 or 30 individuals. And then I talked to Scott Hammer subsequently, who does AIDS vaccine clinical trials, and he was encouraged as well. And he said, we'll never repeat this, but we can go on with what we learned.
Yeah.
So it's great. So that's a big deal.
Yeah, and it suggests that this type of dual vaccine approach, it was a prime boost approach. You give one vaccine as a primer, and then you give the second as a booster shot. And the canary pox vector was used for the primer, and then the booster shot was just a GP120 antigen, I think, right?
Yes. Purified antigen.
Right, purified antigen. Yep. And again, neither one of these had worked in isolation, but it suggests that this general type of approach might really be the way to go, and that's really the direction that I think the whole AIDS vaccine community is now going to be looking.
It's a good thing they didn't use an adenovirus vectored antigen.
Yeah.
That's running into trouble.
As we discussed, I think you guys were both on the TWiV where we talked about that. The adenovectors caused the failure of one of these trials.
Yeah.
And in fact made people more susceptible to HIV than those who didn't get immunized. So they were fortuitous in choosing that combination.
Yeah.
So more of these are ongoing, and we'll hear more about that in the next year as well. And that's why we picked this one. It's a big deal, and it's also ongoing.
Very big deal.
Very difficult problem.
Yes.
AIDS vaccine. I mean, I can't think of a setup that has more obstacles for creating a vaccine in this. It's really it's a horrendous problem.
Yeah, I'm glad you mentioned that, because we have a lot of viral vaccines in particular. They're not easy to make, but they're there and they work. Polio, measles, mumps, rubella, etc. Yeah. And this one is confounding.
And, full credit to the early vaccinologists, Maurice Hilleman et al. They did a yeoman job in producing those vaccines, but really, in retrospect, they kind of went for the low-hanging fruit.
Yeah.
The viruses that we have vaccines against are the ones, not coincidentally, are the ones that were relatively easy to develop vaccines against.
In fact, we picked that biography of Maurice Hilleman way back in May.
Yes. Yeah, that was a pick.
What was the name of that book? Vaccinated. Yes, I highly recommend it.
By Paul Offit?
Yes. Yeah. Amazing recount of all the vaccines that Maurice Hilleman made. I really gained a lot of respect for him.
He had an astonishing career. I interviewed him right before he died, actually.
Yeah.
Very interesting guy.
Where did that interview appear?
That profile was in Nature Medicine.
Okay. So. We'll link to that, because I think that would be great for people to look at.
Yeah.
Story number 4, colony collapse disorder.
There was a lot of buzz about this.
It's right.
Alan's awake.
Yeah, there is. And this one has actually been incubating for a couple of years. Colony collapse disorder is the large-scale unexplained losses of managed honeybee colonies in the United States. They main trait a rapid loss of adult worker bees. So a number of labs have been working on this to try and understand, because obviously honeybees is a big industry in the world, And there have been quite a few papers, and a couple of them came out in 2009. A couple of them have come out beforehand. But many of the qualities of colony collapse made it seem like it was an infectious disease of some sort. And honeybees are apparently infected with a variety of pathogens. And so Ian Lipkin's lab did a metagenomic approach to looking at what kinds of microbes might be involved. They sampled hives from different locations and then used metagenomics. They sequenced the pathogens that were in these bees. And in the Lipkin study, which was published in Science, one organism, Israeli acute paralysis virus of bees, was strongly correlated with colony collapse disorder. And that's a picornavirus-like virus. It's related to poliovirus. And it causes paralysis of bees. So how it would cause the colony collapse disorder is not clear. Because, in that disorder, the workers disappear.
Well, if they got paralyzed out in the field, they would get eaten.
Yeah, but then you would suspect that some would get paralyzed in the hive as well, right?
You would, yes.
Yeah, and apparently there's not a lot of dead bees around the hive, right?
Right.
That's one of the things I saw really interesting. Maybe they get lost. Navigation is a big deal for these.
It is, but as you know, there's always going to be some in the hive. So, so far, I haven't seen that has stood up to be the main cause. There's got to be something else. And now there have been other studies, and there's one published in August 2009, Changes in Transcript Abundance Relating to Colony Collapse Disorder in Bees. So in this study, they did microarrays of RNAs in the bees to try and find out if there's a pathogen involved or the host is responding in some way that would indicate an immune response. Overall, they found elevated expression of pesticide response genes was not observed, so one possibility was that this was a pesticide-induced response. Genes involved in immune response showed no clear trend in expression pattern, despite the increased prevalence of viruses and other pathogens. So they also found more viruses and other pathogens in these colonies, but no indication that they were doing anything. What they did find was unusual ribosomal RNA fragments that were conspicuously more abundant in the guts of CCD-bees. And so they suspect that fragmentation of ribosomal RNA may be part of the immune response. And I think Rich was on a TWiV when we talked about this, and you suggested that it would be RNA cell, which is one of the...
Yeah, the one place in my personal experience where I've seen this before is in certain circumstances in poxvirus infections when apoptosis is induced,
right.
And that can be through the RNA cell pathway. You get apoptosis induced that includes induction of RNA cell, the ribosomes break down. You get discrete fragments of ribosomal RNA, and then because the vaccinia polymerase is pretty promiscuous and will basically polyadenylate any old 3' end lying around, adenylates those things. And that's one of the things in this study with colony collapse disorder, the way the experiments are done implies that the RNAs that they're looking at, fragments are actually polyadenylated. So you not only have to explain the fragmentation, but the polyadenylation. At any rate, I've seen this before under circumstances where the antiviral response is induced. And so that makes me think that maybe what you're seeing in this case is, in fact, an antiviral response. They had a lot of other to say about ribosomal RNA degradation that they had lots of different ideas that were new to me.
Yeah.
No, and another thing to keep in mind, though, with the CCD story, and this sort of came out later toward the end of the year, was there's been a lot of speculation that maybe what we're seeing is just the result of these commercially reared hives being cared for under very, very artificial conditions. They're trucked thousands of miles across the country. They're fed when they're not actively feeding on flowers. When they're being transported, they're fed an artificial nutrient mix of basically high fructose corn syrup. So they're leading very, very strange lives for honeybee colonies, and that could be stressing them and predisposing them to a wide range of problems.
Sure. No, I think that's a good point. And maybe a chronic fatigue syndrome-like disorder where there's some kind of stress leading to infections with a variety of microbes, who knows, and it's showing up in these microarrays.
Right, because apparently it's not. It's either not a problem or not a significant problem for hives that are just privately maintained in people's backyards.
There is some evidence that this is infectious, however, right? I mean,
right.
You can there are such things as contaminated hives.
Yes.
You take
yes.
naive bees and they'll catch the disease.
Yes, that's right. There is an infectious component.
Right.
So our discussions of this attracted a Tom from California, who is a honeybee breeder.
Yeah,
okay? This
was great.
So he's interacted a couple of times, and so I asked him about this, because this has been a big story in 2009. Now there's a documentary film called Colony, which says that the unexplainable phenomenon known as colony collapse disorder has left landscapes of empty beehives all across America, threatening not only the beekeeping industry but our food supply. So I asked Tom, is there a pandemic that will wipe out the world's honeybees? And I put up his answer at Virology blog, but let me just read you a couple of things. Here's his answer. I can only give you my opinion about what is happening with bees today. There is a lot of confusion and misinformation, much of which has been caused by all the media attention over the last several years. As I'm sure you know, the press always seems to get things wrong and emphasizes the doom and gloom crisis aspects.
Hey, it sells.
Sounds familiar.
Yeah, if it bleeds, it leads.
The fact is that there are many more problems that the bees have to contend with. Globalization has brought a steady stream of pests and diseases, and over the past 40 years that I've been keeping bees, I've had to deal with these problems: mites of all kinds, viruses, microsporidians, African bees, small hive beetles, increased pesticide use. So he says basically we have a lot of problems, and as a consequence, many beekeepers have given up. The number of managed hives has decreased from 5 million in 1950 to just over 2 million today.
That's really significant. That's a big deal.
Yeah.
Also, the number of feral colonies has gone down. It's interesting. So wild bee colonies. So he says he doesn't believe that they're in any danger of going extinct. We have all kinds of problems, but in fact, the attention is good because they get more money to do research, but we're not on the verge of dying out. He's happy for the attention, but doesn't think they're going to go away anytime soon. But I think this is a neat story, and it's something we're going to hear more about in 2010 for sure. I don't know if we'll have answers, but we will, as usual, keep you on top of it. So that was number 4.
It'll be interesting to see what happens, Yep.
Okay, next, number 5, AIDS-like disease in wild chimpanzees.
Yeah, I like this. I really like this story as much for the methods as anything else. I mean, it's really
yes.
It's really a heroic effort by these people to understand the sort of natural transmission and pathogenesis of an immunodeficiency virus in a natural host in these chimps. So the background here is that there are something like 40 different immunodeficiency viruses, HIV-like viruses that infect apes. And the dogma has been that these don't cause disease in these animals. They simply carry these viruses, and it's an inconsequential infection. And I think that's where the other bit of interest in this story comes in, because as we've discussed many times before, one of the courses that a virus infection or a host-pathogen relationship can take over time is to establish an equilibrium where the host is resistant, the pathogen is less virulent. And by looking at these infections in apes, one might be able to get some idea of, maybe where HIV-induced disease might go somewhere out in the future in humans. The thinking was that the retroviruses in apes were at some sort of an equilibrium, didn't cause disease. So the bottom line on this study was it shows that the simian immunodeficiency virus that infects chimpanzees, in fact, does cause disease. This is like a huge study. It's over at least 9 years, though I think she's been studying these things for a longer period of time. These are the Goodall colonies, right? Or some of the ones that Jane Goodall studied in Tanzania, near Lake Tanganyika, and she follows 100 chimps over a period of 9 years. And what fascinates me is that the analysis has to do with assaying their urine or feces. Turns out feces is the easier way to do this. You can collect droppings, and at some point or in time, you can identify what animal a dropping came from. And at that point, you can, by sequencing mitochondrial DNA in the feces, in the future, identify which chimp is responsible for this dropping. So you don't even have to see them. You can just collect the feces and figure out which animal was responsible. And then they can also look for actually antibodies to the virus, and they can look by PCR for nucleic acid to the virus. So they can look at the rate of infection in the colony, and they can ask about the fate of the animals. And basically what they found was that the apparent death rate in animals that were infected was significantly higher than the apparent death rate in animals that were not infected. Apparent is important here because animals just disappear. And at some point they have to sort of make the assumption that the animal has actually died. But in a couple of cases, they were actually able to follow animals pretty closely and do autopsies, and were able to basically characterize the progress of an immunodeficiency-like disease and do the pathology when it was all over, and show that they apparently died of something that looks very much like HIV. So the bottom line is that these animals, contrary to what was previously thought, actually get disease from the chimpanzee SIV. And the implication is now there's been a decline in chimpanzee. They're wondering if it could be in part responsible for that decline.
Yeah, sure.
I don't know. So I thought that was a really interesting study.
It's amazing methodology. You're right.
Yeah, there's a whole slug of authors on this paper, and you can't help but picture them. As a matter of fact, I saw her she gave a talk at the American Society for Virology meeting in Vancouver, and they had pictures of people doing these collections and the analysis. And the motivation that has to go into a study like this is just phenomenal.
Yeah.
It's amazing.
Puts us to shame just sitting in our labs doing plaque assays, Rich, you know?
Right.
That's great. Okay. That's number 5. Number 6 is very cool. Diverse viral communities in an icy Antarctic lake.
Extremely cool.
Yes, very. This is a great story. Now, I've spent my whole virological career studying one or two viruses in incredible detail, right? And I'm really attracted to these studies where people go out and find new viruses in extreme places. I don't know, maybe it's because I've had such a focus my whole career, but this is an example of such a study. That we've talked about viruses in the oceans, but this is a freshwater lake in Antarctica surrounded by ice, and they wanted to know what's in these extreme environments. There's not much life there. There are very few more microorganisms, but there are some. Low temperatures, low nutrients, and dark for most of the winter. What kind of viruses could possibly live there? So that's the question they asked. And they just sampled the water in the spring when the top was thawed, and also in the winter when it was frozen over. They drilled through the ice and they sampled the water and filtered it and sequenced it to look for viruses, basically. And they find an amazing diversity of viruses. They take some pictures of them so we know what they look like. They get viruses from 12 different families. And some of them are brand new viruses that we have never seen before. And in addition, the ones they find when the water is not covered with ice, they call the spring virome. The spring virome. This is Lake Limnopolar, by the way, in case any of you want to go there. The spring virome is dominated by single-stranded DNA viruses, little guys, circoviruses, Gemini viruses, nanoviruses, and then, but they're not only phages. They also are viruses that infect mammals, birds, and plants. Now, There aren't any mammals, birds, and plants in this lake, so they must be infecting something else.
Some other eukaryote
Yeah.
that's swimming around in the lake, I suppose.
Yep.
Or something that defecates in the lake. Yeah.
They talked about how there is access to the lake from waterfowl and stuff, but they tried to minimize that in a way. But that's a possibility.
Yeah.
Yeah, because if the virus, I mean, these viruses are not necessarily actively infecting anything in the water. If you have, I don't know, penguins pooping on an iceberg several miles from there, and then it drains down into the lake, then you could end up with the virus preserved in this ice cold water sitting there.
Speaking of penguins, I got to take a diversion here. I bought this Planet Earth DVD that Rich and Dick were going crazy about a few TWiVs ago.
Right.
And it begins with the penguins down in the Antarctic during the winter.
Right.
They're all huddled together because it's bloody freezing.
Yeah.
They
That is not a good time.
Make a huge mass of huddled penguins, and a few peel off now and then to do stuff, but it's amazing.
Have you seen March of the Penguins?
No.
Yeah,
that's a whole video entirely devoted to that phenomenon, and it's amazing.
Do we know anything about the viruses in penguins? No.
Well, I don't.
Amazing. Someone might, but they could be shedding into these areas as well.
Yeah. If you want to know what's involved in researching these, the classic text on it is called "The Worst Journey in the World."
Oh yeah.
Yeah, it's was a guy who was on the Scott expedition to the pole, and he journeyed out to the nesting grounds of the emperor penguins. It's a fascinating read.
Well, that whole Planet Earth thing is amazing. They go all over the place.
It is an amazing series.
They get really animals doing stuff, and it's all in high definition. I bought the Blu-ray version. It's amazing. So you guys are absolutely right on about that one.
I gave it to each of my kids for Christmas.
Yeah, well, I bought it this week, and I sat the kids down, and they were mesmerized for 2 hours.
I'll have to check this out.
You have to check that out. It's really, really amazing. Anyway, so then when so we have the spring viruses, which are single-stranded DNA. Then in the summer, let me clarify this: spring virome and the summer virome, where everything's thought out. I guess they can't go in the winter. It's really too extreme. So they go in the spring when there's ice on top, but it's spring, so I guess it's hospitable. And then in the summer when there's no ice. And those are totally different viruses. They're all double-stranded DNA viruses, big viruses like the mimes and ficodinoviruses and the large guys. Totally different population, which suggests that there's different things supporting them in the two different times of the year.
They need to train a couple of penguins.
That's right.
Go down here and do this.
Sample. They assembled 25 circular DNA genomes with no similarities to known sequences. That is amazing to me. 25 brand new viruses.
This guy in the I'm looking at the electron micrographs, and I'm fascinated by this guy in the lower right-hand corner that looks kind of like a teardrop.
Yeah.
Or seasonally a Christmas ornament or something like that. I've never seen anything like that.
Yeah. I've seen those before. Actually, there's a picture in our textbook. I forgot what that is.
I don't know if I got the rest. It almost looks like there's is there some sort of base plate structure in the upper left point?
Yeah, it does. It does look like it. Yeah, I can't quite see it. There is a base plate. I see. It's different from the rest. Very interesting.
Yeah.
So anyway, this is very cool stuff. Brand new viruses, different according to season, and in a very extreme environment. So what are they infecting? What are they doing there? Lots of interesting questions. The only concern I have is I don't know if we're going to have money to do all the cool things that need to be done.
No, I think you get to do that once.
Because you're discovering lots of viruses. Each of these could be studied separately, but there's not money to do this.
Right.
And, there are other extreme environments that can be looked at and should be looked at, but...
And if you really want to study these, you need to figure out how to culture them, which means you need to figure out what they're infecting.
Right. But massively interesting problems here.
Really
so interesting, and I love this stuff, so that's why I picked that, and I think we'll hear more about that next year. All right, number 7 is the Cerro Epidemiology of Polyoma Viruses in Humans.
Right. So this studies the distribution in the... I just closed the article.
I've done that.
That's okay. I can get it back. It studies the distribution in the human population of a bunch of different polyoma viruses. Now, classically, so polyoma viruses are small circular DNA viruses, naked, capsid. They're ubiquitous in nature, infect a bunch of different species, don't cross species very well. The first two to be really studied in any detail were Polyoma virus, which is a mouse polyoma virus, and SV40, or simian vacuolating 40 virus, which is a monkey virus. And so the human viruses that had been discovered in the 70s were BK and JC. Oh, and I should say that the interest in these, they're really, under normal circumstances, this wasn't understood immediately, but under normal circumstances, they're really pretty benign. They don't do much. You acquire them as a respiratory infection, and ultimately they migrate most often to the kidneys and establish a persistent infection in the kidneys, and don't really cause any sort of pathology that we're aware of, except under conditions of immunosuppression. They attracted a lot of attention in the 60s because they encode proteins that go by the general name of T antigen for tumor antigen. These are proteins that regulate the cell cycle for the purposes of the virus infection, but as a consequence of that, under special conditions, they can cause tumors, though there's no evidence that they actually do that in nature. At any rate, so recently, over the past several years, some new polyoma viruses of humans have been discovered. There's KIV, WUV, MCV. A couple of these were from respiratory samples from people who did have a respiratory infection, but the association is not clear. MCV came from a Merkel cell carcinoma, which is a rare skin tumor, and that was actually there was an integrated viral DNA in that tumor. And the question simply is, what's the normal distribution of these viruses? And what was found was that they're present at a very high prevalence in the population. BKV, 82%, JCV, 39%, LPV, that's a related one, 15%, KIV, 55%, WV, 69%, and a couple of there were two different strains of MCV ranging from 25 to 42%. And they found that this is all done by looking for antibodies to these viruses in the population. They also found that the antibodies showed up in the population at a very young age. So apparently people are getting infected in childhood, and then certainly the antibody response to the infection persists through adulthood. But knowing what we do about polyoma viruses, I would be surprised if the viruses themselves didn't persist for a long period of time as well. What interests me about this is that it comes back again to this idea that there are a lot of viruses out there that really have established an equilibrium with their hosts and don't really do much. You don't see them if Alan said a little while ago, you find what you're looking for.
Yeah.
Right.
And they don't cause any disease, so they don't show up, and you actually have to go out there looking for strange polyoma viruses to actually find these. And there's going to be many, many more viruses out there, I think, that we just live with. And that's just fine.
And you know what would be good? Now, here they make the antigen. I don't remember they look for antibodies to each one, right?
Right.
So if there were only a way to just look directly for viral antibodies without having to make antigens and tell if they're against, there's no way to do that, unfortunately, at the moment. Right. But wouldn't that be the way to do it? So you don't need to have an antigen for every virus that you're looking for.
Right. And the other thing that wasn't clear to me in this paper is the extent of co-infection with these different viruses. Maybe if I look more closely at the data, I could find it. But with the kind of infection rates they're talking about, it must be that a lot of people are infected with several of these simultaneously.
Yeah.
Really interesting. Now, the other bottom line out of this is the SV40 issue. I know that you've talked on the show before about the fact that one of the first polio virus vaccines was contaminated with SV40 virus. And since this is a monkey virus that in culture under artificial conditions can transform human cells, there was fear that shooting up the human population with SV40 might cause problems down the road. And so there's been interest in finding out how many people really are infected with SV40 and whether there's a problem. And what these guys find is that the apparent seroprevalence of SV40 is down around 2%. They don't even think that that's significant, and they can't find any evidence from this that SV40 actually infects humans on a regular basis or causes any kind of problem.
Yeah, I think that's an important kind of hidden point here.
Right, because that's been very controversial.
Absolutely. A lot of lawsuits ongoing about that at the moment.
Now, having made that point, you still don't want extraneous viruses in a vaccine, even if they turn out to be harmless later.
No, that's right.
No.
That's right.
But in this particular case, it does not seem to have been the end of the world.
Right. No, I agree with that. But, we probably have Xmrv in the blood supply right now.
Yeah,
so
most likely.
Who knows? Okay, number 8 is the UK red squirrels being threatened by pox virus.
So I like this because once again, it speaks to this sort of equilibrium issue and the natural history of virus infections. I also like it. Well, I like it because the squirrels are cute. Okay? The red squirrels are cute. And I also like it because when it came up, superficially, it looked like a pretty simple story. And then as you got into it, you realized that it was much, much more complicated and much, much more interesting. So the idea here, the issue here is that there are red squirrels that are common to the British Isles and Europe, and gray squirrels ordinarily live in North America. But gray squirrels were introduced, and it's not clear how, into the British Isles over 100 years ago in the late 1800s. And what has been noticed is that where the gray squirrels are present, the red squirrels decline in population. And it's also noticed that the red squirrels get a poxvirus disease called Squirrel Pox, which once sequenced, we find out that it's a poxvirus that's out there on its own branch of the poxvirus phylogenetic trees. So the interesting thing about this is that it turns out that the poxvirus doesn't do anything to the gray squirrels. They're carriers, or it does very little. They've done experimental infections and saw no clinical symptoms at all, but they are probably the reservoir, and it's absolutely lethal in the red squirrels. You can tell this both in the natural situation with experimental infections. If they get infected with this virus, they die. So the gray squirrels are harboring this virus and killing off the red squirrels. Now, the interesting thing about this is that this is, as we pointed out, really similar to the classic myxoma story, where there's another pox virus that is in equilibrium with a South American rabbit, but kills European rabbits and was used as biological control for the European rabbits that had overrun Australia. And over time, what happened was that the infection in the Australian rabbits reached an equilibrium where the rabbits were more resistant and the virus was less attenuated. And you have to wonder whether the same thing might ultimately happen in this case. But I was thinking about this, and the circumstances are really different, because in the myxoma case, the susceptible rabbits were actually carrying and transmitting the disease. And in this case, the gray squirrels are the primary reservoir. And so the virus probably has to maintain certain properties in order to maintain itself in the gray squirrel. And that's going to affect this whole, I would think, this whole evolution of virus types.
Mm-hmm. Right. There's not a strong selection for the virus to not kill the red squirrels.
Exactly. At any rate, let's hear it for the red squirrels. I hate to speak against my, Yankee gray squirrels, but those guys are really cute, and I hope that they manage to make it through this somehow. But they've got a struggle on their hands.
That was TWiV 63. Well, we'll follow them in 2010. See what happens. Yeah, that's a great story, too. I love it. Number 9 is a story that we did on TWiV 29 back in April, I think. Polio spreads from Nigeria to 15 African countries. And this is actually a story that is ongoing. It's about the eradication of polio by WHO, CDC, and many other organizations.
Now, they completed that in 2000, didn't they?
Yes, of course. And I've destroyed all my polio stocks and no longer work on the virus. That's not true, actually.
Sorry, we're being a wee bit sarcastic.
Silly. This story actually begins in 2003 when Nigeria decided to stop immunizing its population against polio. Polio, of course, there are 3 serotypes, all of which cause polio and all of which are included in the trivalent vaccine. So they stopped immunizing for a year, and of course polio spread throughout Nigeria and to other countries as well. The next year they decided to start immunizing again, but the consequences of that interruption continued to this day, and this is an example of that. There was an outbreak in Nigeria early in 2009, and it spread to 15 other countries, many of which had not had polio for some time. It just goes to show what can happen if you don't keep up the immunization efforts. Now, right. The interesting other aspect of this story has to do with the fact that when Nigeria resumed immunizing in 2004, it was decided that they would use two monovalent types 1 and 3. In other words, 2 different type 1 vaccines. These are both live and infectious vaccines that are taken orally. Because in 1999, poliovirus type 2 was declared eradicated from the globe by World Health Organization. So there was no reason, it was thought anyway, to keep immunizing people in Nigeria, so they went in with only types 1 and 3. Well, type 2 polio reemerged in Nigeria late in 2005, and it was actually vaccine-derived type 2 polio. Not wild polio, but polio derived from immunization of individuals. So throughout 2009, there has been activity of type 2 polio in Nigeria, as well as types 1 and 3. Just last month, Afghanistan began to use, this month, December, I guess that is last month, yes. Yeah. Afghanistan began to use a bivalent type 1 and 3 polio vaccine. So not separately as they did in Nigeria, but the 2 of them combined. And this has never been done before. And I'm a little puzzled by that because type 2 vaccine-derived polio is still around. I mean, it was isolated.
That doesn't make any sense.
It was isolated in Nigeria as recently as October, I believe. So even if it hasn't been detected since then, it doesn't mean it's not there. And that's the trouble they got into in Nigeria in the first place with type 2. So it seems to me this is asking for trouble to just use a bivalent vaccine. Right. And my prediction for 2010 is that we're going to see type 2 polio in Afghanistan. I don't want that to happen.
But it seems like they're setting themselves up for it.
Don't they?
Yeah.
I mean, it's they say on the polio eradication site, wild type 2 polio has not been seen anywhere in the world since 1999. Yes.
Well, who cares?
Right. The vaccine-derived strain will still paralyze your kids.
Exactly.
And this is an inherent problem of using the live polio vaccine. This is not true of all live vaccines, of course, but in the case of the polio vaccine, the live vaccine can revert to the paralytic virus, and this is an inherent problem in using that vaccine for an eradication campaign.
Yep.
So the solution to the eradication problem is to use the inactivated vaccine, right?
That's right.
Yes.
I think the sooner we transition to inactivated, the better off everyone is going to be.
Gosh, don't you wish somebody had suggested that years ago?
I think we are remiss for not having done so, Alan. Maybe we should write an article.
Yeah, maybe that would help.
I think you already did it, fellas.
Yes.
Sorry, I don't mean to be sarcastic. But what's interesting about this article in Polio Eradication Initiative, which is polioeradication.org, it's 15th December. They don't mention type 2 vaccine-associated polio. They just say, this is a brand new bivalent vaccine. It's great. There's no type 2 around. I think they should be honest about the fact that there could be vaccine-derived type 2 around.
Yeah, I think there's a lot of selective blindness in the polio eradication campaign. I mean, I'm all in favor of vaccinating and controlling the disease, but I think this focus on trying to eradicate one particular virus has blinded them to the reality of the situation.
So I think that is a bad policy decision, and we're going to certainly hear about that in 2010. And we'll keep on top of that.
So the barrier to using an activated vaccine is really economic. Is that correct?
Yes.
It's economics and scale. It hasn't been scaled up yet, and it's not clear if it would work in certain areas because it hasn't been tested in a lot of places where it needs to be used. Now, the last areas with polio are Nigeria, Pakistan, Afghanistan, and India. And it's never been shown that the inactivated vaccine works in those areas. So there's some concern, but I think they have to just start small and work up.
And the thing to do, I mean, the reason people focus like a laser on a polio eradication effort is that this is the kind of thing, you could win a Nobel Prize for and build a whole career out of, that you're going to wipe out this one virus, and it's theoretically achievable. But it would be better, I think, to spend those resources on the much less glamorous job of building a whole health infrastructure, which is going to take a lot longer, and it's not going to be the glorious conclusion. But that's really what you need, is an infrastructure where you can deliver not just oral polio vaccine, but measles vaccine and all the others that just isn't on the agenda as much, I guess.
All right, number 10, how mosquitoes survive dengue virus infection.
Yes, and we think about dengue virus infecting humans, but who cries for the poor mosquito? Well, I certainly don't. But in any case,
I think Dick would if he were here.
Yes, Dick would.
Yeah, he would.
And I'm a bug guy too. I do like a lot of insects. They're fascinating creatures, but I think if the mosquitoes vanished from the earth, I would not shed a tear. But in the case of vector-borne disease, it's important to look at the whole biology and the ecology of the disease. So what these folks at Colorado State did was they looked at dengue type 2 infections in the mosquito vector for dengue, Aedes aegypti. And the question was, what does the virus do to the mosquito? We know what it does to the humans. Does it kill the mosquito at any appreciable rate? Does the mosquito mount an immune response? And it turns out that the mosquito does seem to mount a sort of an immune response. And mosquitoes, insects in general, don't have the same immune system that mammals do. But they do develop a short interfering RNA, an RNAi response that's triggered by double-stranded RNA. And double-stranded RNA, of course, occurs when you get a lot of viral infections, including a dengue infection. So they looked in the mosquitoes and they found that there were these interfering RNAs that were produced in response to the dengue infection. And these little RNAs are complementary to the dengue virus genome. They should inhibit the virus replication. And interestingly, the mosquitoes still get the infection, and the virus appears to be able to circumvent this response. But if you inhibit the RNAi response in the mosquitoes, the virus replicates much more efficiently. So this is part of an ongoing biological war between the mosquito and the virus. The mosquito is responding to the virus. It is inhibiting the replication, although not completely. And the virus, in turn, has evolved its own responses against this RNAi so that it's still able to replicate. But it's typical of immunological stalemates that we see in humans. You get a certain amount of response, but the virus has ways of evading it. The same thing is going on in mosquitoes. And the really interesting thing here, I think, besides the issue of looking at the whole ecology of the virus, is that the virus and the mosquito have reached this evolutionary stalemate, as often happens with an immune response. You get the host immune response. In this case, the mosquito is generating the RNAi, and then the virus responds to the response and has a way of circumventing it. And that's exactly what we see in most human infections that we think about, and it's what we see in this mosquito when it's infected with dengue. So I thought this was a really interesting study for a couple of reasons. First, because they bothered to look, that they're really paying attention to what's going on in the vector. And second, that they found something, that they found a mosquito immune response against the virus and in turn a viral response against that. So it suggests, maybe if you could manipulate this pathway somehow, either chemically or biologically, you might be able to impede the viral replication in the mosquito, and that might help interrupt the chain of transmission in the humans as well.
Yeah, this is. I wonder how general this is going to be. I wonder if the gray squirrels do something similar.
Well, there's going to be some sort of equilibrium that's established. The pox viruses are notorious for making immunomodulatory gene products, and so I'm sure that equilibrium has to do with a long-term interaction between the immune system and the host and the virus to establish this sort of equilibrium, and it doesn't play out the same way in the red squirrel.
Sure.
And the RNAi response, I mean, mammals we now know make small interfering RNAs for a variety of purposes, and I'd be surprised if those were not involved in at least some antiviral responses.
Sure.
So this appears to be an evolutionarily ancient immune system, and presumably it's still going on in us, and maybe this similar type of thing is happening in human cells.
I think, as you said, whether we can manipulate this in some way to control mosquito-transmitted diseases would be very interesting. I think it's a ways off, but this gives us some clues.
Yeah. In the meantime, just put up window screens.
That's right. Okay, those were our 10 stories, and I'd like to do just a couple of emails, but before we do that, I wanted to just summarize TWiV for the year and tell you where we're going. We had 51 episodes in 2009, including a special with Peter Palese. We did one live show at the American Society for Microbiology. We tried to do a live video stream, and we experimented with some screen video and as well for our Virology 101. And thanks to our listeners, we're still on the featured page at iTunes. If you go to the podcasting sections in iTunes, Science and Medicine /medicine, TWiV is usually in the featured central panel. So we thank everyone for being part of TWiV and allowing that to happen. We also began TWIP, our parasitism podcast. We have 3 episodes so far, and we've been up as high as number 4 on the top podcasts in iTunes as well.
Nice.
Pretty good. So I want to thank everyone for listening and participating with email questions, suggestions, corrections, and everything. Keep them coming. And also thanks to the American Society for Microbiology, Chris Condayan, Ray Ortega, Garth Hogan for their help in helping us in promoting TWiV and TWIP. For 2010, we have some interesting things happening. We're going to have, of course, more new guests. We'll be talking about things that you've suggested: prions, papilloma viruses, herpes viruses, evolution, and lots of other topics. We're going to experiment with video. A company named Viomed.com is going to live stream and record one TWiV each month starting in January. So they're going to send a video recorder over to the TWiV studio in New York and stream it. I'm not sure it'll be so interesting in January because Dick won't be around, so it'll just be me in the office. But if he's there in future months and other guests, that'll be interesting. Oh, yes, we're going to be at the American Society for Virology meeting in July. That's going to be in Montana. And hopefully we'll let lots of virologists learn what we do and what can be done with this medium. I'm going to be teaching a new virology course in January at Columbia to undergraduates, and I'm going to be posting all those screencasts online. And finally, we have a new sponsor coming this month, and that'll be announced next time. And we are going to have a giveaway contest of the product that the sponsor makes. In fact, we're going to have a 3-month giveaway. We're going to give away one of those items every month, and they are very cool. So stay tuned for information on that on our next show. Okay, let's do a couple of emails and we'll wrap it up. Cedric writes, "Hey, Vince, Alan, Dick, and whoever else is joining you this week. I love the show and I hope you keep up the good work. I'm a biology student with diverse interests in science, and I enjoy the weekly dose of virology I get from the podcast. I have a couple of questions I would love to hear you discuss. First, you recently read an email from a student who had just finished a physics degree and was planning to go to graduate school in biophysics. He asked about how you all balance the vast amount of information in your field and other fields of science You did not really address this, and I was hoping that you would. As a current student, this is something I often think about. One of the things I love about biology is its breadth, but this also presents a challenge for students seeking a well-rounded and comprehensive understanding of biology. Biologically relevant topics range in scale from small molecules like water to global ecosystems. Biology offers lots of opportunities for interdisciplinary work, as knowledge from chemistry and physics, geology, astronomy, and even social sciences can lend themselves to biological problems, and discoveries in biology lend themselves to other fields. You guys seem to have a wide range of interests in biology and the other natural sciences, so I'm wondering how you keep current. Also, what advice do you have for students? When I thumb through my course catalog, there's no shortage of classes in the physics, chemistry, and geology departments that sound interesting, not to mention all the biology electives. What recommendations do you have for a student to balance the breadth and depth about that biology offers?
Ask other students who the good teachers are.
Ooh, good idea. Wow.
Because it's not. You could take a geology course at one university that is just absolutely fascinating and completely revolutionizes your view of your own field, and you could take the same course at another university and you'd say, geology, whatever. I think the teacher is what makes the course, and probably the best course I had in my entire college career, before I got to graduate school, was organic chemistry. And it was absolutely amazing. And I talked to other people who are biologists who say, "Oh God, I hated organic. It was a chore." And it's entirely a function of the professor I had, who was outstanding. So I would say, don't focus so much on the subject matter when you're looking at the course catalog. Ask around about, is this course enlightening? Is this teacher really gonna blow my mind?
What about you, Rich?
That's really I can't do better than that on choosing courses. I think that's just terrific advice. Beyond that, I would just say, obviously there's going to be if you're focused in a certain field, there's going to be a certain amount of core curriculum that you have to deal with. And beyond that, I think it's really important to sort of follow your guts on this, and if something looks interesting, and if you do Alan's research and there's a teacher out there who's good, just go for it. Don't be shy about it. Cultivate your own diversity that way. With respect to keeping up, I'm actually not a good person to ask. I've been, over my lifetime, really not very good at sort of methodically reading the literature both in my field and out of my field. But what I've discovered is that as time goes on, it takes care of itself. I would say my first bit of advice is to be patient, because this knowledge accumulates slowly. And the more knowledge you accumulate, the better the new knowledge sticks, because you create a context. And just be patient, and it will happen. And then what happens is somebody asks you to teach something. That's when you really learn it, okay? Or somebody sends you a paper to review, and that's you learn what's in the paper. Or somebody asks you to sit on a study section, or best of all, somebody asks you if you'd participate in a TWiV. I've learned more this than anything else. And then beyond that, of course, there's all the conventional stuff. Nowadays, it's great. You can subscribe to a number of services that send you selected publications in areas of your interest, and you can thumb through those and see what you like.
Yeah, that's true. And don't let yourself get swamped. If you find something is starting to bore you, then read something else.
Absolutely. I agree with that completely. I was going to relate a thing. I used to go to seminars in graduate school and come out of the seminar, and everybody was going, "Oh, that was terrific," and telling me all about it and stuff. And I'm thinking to myself, is there something wrong with me? Because I thought it was boring. And I really didn't get it. That's going to happen. Don't worry about it. It's okay not to be interested in something. Just follow what you're interested in.
Yep.
I'd also add, take the courses you have to take, find the great teachers, as Alan suggested, but also try iTunes University on iTunes.
Yes.
You can find lectures from all over the world there in all fields, and you can find the best ones.
Yeah.
And if you can't take an interesting geophysics course, you can just listen to the podcast. And I'm telling you, some of them are videos. They can be just as good as taking the course.
Yeah.
So you can really broaden yourself. And I have broadened myself by doing podcasting. There's no doubt about it.
Yeah, it's been tremendous for me.
Talking to Dick Despommier, for example, who has totally different interests in some ways, and in Alan as well, you learn. And if you can listen to podcasts, iTunes U or any others, unfortunately, there are not a lot of hard science podcasts, but you can find some others in other areas, especially there are a lot in astronomy. And I would just do that to complement your courses. All right, his second question, what do you wish all biologists knew or appreciated about virology? The question is inspired by the microbial ecology course I'm taking this quarter, which is taught by 2 microbiologists. One thing I will be taking away from this class is how underappreciated microbes sometimes are, even by other biologists, and how the diversity of bacteria and archaea is not always given the attention they feel it deserves. We would all love for non-scientists to have better understanding of biology, but I'm wondering what aspects of virology you feel deserve more attention in introductory biology courses, or what you wish that biologists in other fields understood about viruses. That's a good question.
Yeah, very good.
Well, I think the greatest thing that's not appreciated is how viruses evolve, and that they can be studied better than almost any other organism because they have such diversity and such huge numbers. And when you get past that, then you can see evolution happening within a day in a cell culture. And I think that is underappreciated. And it's not easy to get to that. It took me many years to really get that. And I think if other biologists could get there, they would learn and understand a lot more about viruses.
I think a couple of things. One is, I don't think a lot of people appreciate that viruses have their own family tree. You know, it's not just something that replicates in a host. You got rhabdoviruses that infect species from 3 or 4 different animal parts of the animal kingdom. Okay, they've really evolved as their own evolutionary entity with their own family tree, and I don't think people really fully appreciate that. The other thing that I emphasize to medical students or others that I teach is that they're tricky little suckers. Okay, you can try and generalize about viruses, and really, in the end, the only generality you can make is that they all need to make messenger RNA, and beyond that, for every rule there's an exception. For every defense that the host has, there's an offense from the virus, and they are very, very tricky and very diverse in the tricks that they use.
I think the thing that I would those are excellent things to bring out. I think I would wish that more biologists understood just how elegant viruses are, and when I say that, I mean in terms of the amount of information and the lack of extraneous information that they carry with them. And you can point to examples, certainly like Mimi viruses, which could, appear to carry an enormous amount of extraneous baggage. But in general, I think virology represents biology at its in some sense most simplified extreme. You look at the genome of a virus, and in many cases there is just pure information. There's no waste. There's everything is maxed out in terms of the information that this virus can carry into a cell, because that's all there is to it. That's all that virus has to go on. It doesn't get to, in most cases, carry a lot of organelles around with it. So it is a highly evolved, elegant system that has evolved for a specific goal, and that goal is actually the goal of all of biology, to reproduce itself.
Yeah. Yep. And one more thing, viruses are not all bad. Some of them do good.
Right.
I think that's a misconception that all viruses make you sick, despite the tagline of TWiV. It ain't true. All right, let's do one more from Soraya. Dear Professor, we have just published a picture book for kids that goes through the mechanism of viral infection and contagion, which is also available on our website for viewing free of charge. The link is, and we'll post it in the show notes. You will see that while some basic things are quite accurate in concept, it is a fictionalized book that we hope will have a wider appeal to 4 to 8-year-olds than one that is purely didactic. We are seeking contributions from scientists that can be used to enrich our website and to contribute to a companion activity book online activities in return for free copies of the book. Should you have ideas for how we might get the word out to the virology community for online groups, newsletters, etc., please do let us know. Well, we have many, many listeners here at TWiV, so we'll post this and have a look and tell us what you think, and we'll give Soraya some feedback. She's an editorial assistant at the company making this book, magicworldmedia.com.
It's a pretty cool concept.
Yeah, it's not bad.
The journey of rhinovirus.
Yeah. I mean, it does fictionalize it, and maybe that's better for 4 to 8-year-olds. I'm not an expert at teaching them, although sometimes I think I am. But listeners, take a look. Tell us what you think and what could be done to make it better. All right, let's do a couple of picks of the week. Alan, what have you got?
I have spaceweather.com. I've actually been following this website for a while. I don't know how many listeners know this, but we've been undergoing a sunspot minimum in recent years, and we have just begun, we have just begun solar cycle 24, and that's not in the history of the sun 24 cycles. It's in the history of people keeping track of sunspots. So sunspots go through an 11-year cycle, a roughly 11-year cycle. You get more of them and more of them and more of them, and then they decline. You get fewer and fewer, and you get a sunspot minimum, and then you go through another cycle. This particular round at the end of solar cycle 23, a few years ago, the sunspots pretty much went away, and they were supposed to start up again, I guess about 2 years ago, and they didn't, which was a little disturbing. Because this is the sun, and it's behaved this way for all of the history that we've been paying attention to it, and now we're not getting any more sunspots. So what's going on? To everyone's relief, the sunspots have once again started up, and they're following their usual pattern. We're getting more and more of them. But that was what got me following spaceweather.com. They also cover all sorts of other things. You don't think of space as having weather, but that's kind of an appropriate way to think about it. It's really the interactions between the Earth and the immediate solar system. So there's a lot of stuff on auroras, and the headline article I'm looking at it today is of course, the blue moon yesterday, which was a blue moon on New Year's, which only happens once in a,
Great. Hey, the solar wind is currently 298 kilometers per second at the moment. Going to have to take off the jib.
This is important to me as a ham radio operator, but it may not. That particular aspect may not interest a lot of other people.
Well, I think if you're going to sail your space boat, you take off the jib,
That's right. If you're planning on sending your solar sailor out, you want to know about this.
This is cool. They have an app to track satellites.
Yes.
Satellite flyby apps for your iPhone. There you go, Rich.
Gotta get it.
Could track the satellites flying over. It's very cool. I'd not seen this.
Yeah.
Very neat. Hey, speaking of space, you guys have to see Avatar.
Oh, it's outstanding.
Yes.
Have you seen it?
On our agenda. I haven't seen it yet. It will happen soon.
Yeah,
saw that in 3D. A lot of fun.
And there is some science in it, so there's a lot of DNA and this and that, so it's good. But that's not my pick. But I just thought since you have a space pick here, I should mention it. That's good. Rich, what have you got?
So I'm going to recommend a book. Surely You're Joking, Mr. Feynman, subtitled Adventures of a Curious Character. It's an autobiography, basically, by Richard Feynman with some help from 3 other authors, Ralph Layton, who was a friend of his, and Edward Hutchings and Albert R. Hibbs. So Richard Feynman was an American physicist who won the Nobel Prize for a bunch of quantum physics that I don't understand. He won the Nobel Prize in 1965. He also worked on the Manhattan Project for the development of the atomic bomb. And I think many people will remember him because he served on the panel that investigated the Space Shuttle Challenger disaster. And there's some famous clips of him showing that if you compress the O-ring from the booster and chill it, that it loses its elasticity, which was basically the solution to the problem. But this guy was out there. I don't know, how else to say it. But he was his own sort of philosopher. And he had lots of interesting things to say about just about everything. And in particular, in this book, he has a lot to say about what motivates his curiosity and how his thinking fits into a scientific perspective of the universe. And this is only one of a couple of different books of this type, and there's also some videos of some interviews. And I think that if you get into this and you like it, there's much more down the road. It was one of my favorites from long ago that I just recalled recently.
Sounds great. I have to get it.
Really good stuff.
I liked Feynman a lot.
Yeah, he's crazy.
Crazy, but good stuff. Smart guy, and can teach. He could teach everybody about physics, too.
Right. I think they probably still, I don't know if they still use his textbooks, but they were certainly state of the art for a long time. I
think we've selected there's his lecture series, which you can buy. I'm not sure what medium, video or audio, but I think maybe Dick has mentioned it before on the show, which is very good. Okay, my pick is also a book. It's a new book by Harold Varmus. It's called The Art and Politics of Science. And I just picked it up this week and begun reading. I haven't finished it, but it's terrific. It tracks his career in scientific research. He was a literature major to begin with in college, and then he got into science and eventually went to medical school, and then had a great career, got the Nobel Prize in medicine, and then went on to be an NIH director, and is now the head of Sloan Kettering Cancer Center in New York. So this guy has bridged the entire field, and this book is all about that, and it's really good. He's a great writer, and it's interesting and timely stuff. So that's The Art and Politics of Science. Very good.
Sounds great.
Yeah.
Yeah, I highly recommend it. I'm halfway through, but it just touches everything. I have to read you this one passage here. This is really good. Because our success, he's talking of his research success, brought prizes, especially the Nobel Prize in 1989, and prizes brought prominence, I became active in the world of science policy. A few years later, in 1993, President Bill Clinton asked me to run the NIH. So there you go. It's pretty simple. You get prizes, you get prominence, and you run the NIH. So it's a little, this guy has done everything. So he writes it in a very, what's the way, this is the way it works,
Yeah,
right.
So I think it's good to see that point of view, which none of us, of course, have.
I would like to have a glimpse into his mind, because he's a special person.
Yeah.
All right, that'll do it for the first TWiV of 2010. Hey, guys, is this the end of the decade?
Yes.
This is the last year now, right?
Well, no, I think the decade ended already, because I think we should just stipulate that the year before 1 AD was 0 AD, as it should have been.
I see. Yeah, there is some
that
controversy.
Fixes the whole problem. Just say that there was a 0 and then...
Okay.
You're going to get some pushback on that, Alan.
Oh, of course.
Not everybody's going to buy into that.
Yeah, because I understand the decade ends at the end of 2010, but...
Yes.
Well, we'll do a 10-year review at that time. How's that?
Good idea. I actually started to think about that. I think that would be a great thing to do.
Yeah, that would require some thought, but that would be fun. As always, if you are a new listener at TWiV, go over to iTunes or the Zune Marketplace and subscribe, and that way you get new episodes automatically, and when you subscribe, it pushes our ratings up. And you can also leave reviews in iTunes and the Zunes as well. We're part of ASM, SciencePodcasters.org, and ProMedNetwork.com. And you should also check out our companion podcast, TWIP, This Week in Parasitism, at microbeworld.org/TWIP. As always, send us your questions and comments to twiv@twiv.tv. Thanks, Rich, for joining us.
My pleasure. I always enjoy it.
We will post Rich's URL, which is too long to mention here.
Right now, yeah, but my picture has a beard on it. That doesn't exist anymore. I got to do something.
You don't have a beard? That's right.
No,
your Skype
like 2
picture.
years ago.
Yeah, well, you ought to change that picture. Yeah.
I need to work on this.
Well, enjoy the rest of your your stay out there in Oregon.
I shall.
And we hope to have you back on TWiV again this month.
Anytime.
We'll work it out.
I'm all over it.
Alan is at dovedocs.com. Thanks a lot, Alan.
Always a good time.
Appreciate you both taking time out on New Year's Day. And thanks everyone for listening. We will be back next week. Another TWiV is viral.